Good afternoon, everyone. Thanks for joining us. Welcome to the Enabling Data panel, integrating genomic and patient data to realize the future of personalized medicine. So we have an hour-long panel, which is great. Thanks to the schedulers for giving all of that time to us. We are going to make this, I think, as open to you guys and the audience as possible. Roundtable type thing. So there's a microphone in the middle. We'll encourage everyone to come up, ask questions, and engage and respond to questions as well. It's not just about us entirely. I think the first thing we'll do is introduce ourselves. My name's Will Salerno. I'm the Director of Genome Informatics at the Human Genome Sequencing Center at Baylor College of Medicine. Do lots of complex common disease sequencing and a lot of Mendelian disease and everything in between. We partner with Baylor Maraca Genetics Labs. So figuring out how to integrate the patient data with the genomic, we definitely are on the genomic side. We have a lot of that to offer and thinking about clever ways to integrate the patient data and translate that back into health solutions.
Hi, thanks for inviting me. I'm Riccardo Sabatini, Data Science Lead for Human Longevity, Craig Sventler Genomic Company. We have one of the largest efforts in full genome sequencing for clinical, let's say, relevant data. We do about one full genome sequence every 15 minutes, 24 hours a day across all the year. What I work on is actually building added value and doing machine learning applied on full genome sequencing. So the quality of the data and the amount of data that we can collect is a critical feature. We presented a couple of models and a couple of papers in the last year to predict common traits, to predict other clinically relevant things. This is one of the assets on which we build, let's say, the clinical service that is the nucleus. More than happy to answer any question and start a conversation.
Thank you, Riccardo. Hi, I'm Kurt Bachman. I'm at Janssen Pharmaceuticals, which is part of Johnson & Johnson. It's the pharma sector of Johnson & Johnson. I lead a group that crosses therapeutic areas: oncology, immunology, cardiovascular, neuroscience, and infectious disease, looking at computational and systems biology approaches, multiple omics platforms, integrating multiple omics platforms all the way from chromatin to proteomics, and linking that with phenotypes, primarily for target discovery and clinical biomarker for early biomarker clinical trials.
Hi, I'm Elaine Johansson. I'm with the Food and Drug Administration. I'm the Deputy Director for the Office of Health Informatics. I'm also the Project Manager for Precision FDA, which is FDA's attempt to develop a community around next-generation sequencing.
Hi, I'm Justin Zook from the National Institute of Standards and Technology, where I lead our work helping to coordinate the Genome in a Bottle Consortium work. Is this louder? Yes. So I help to lead the Genome in a Bottle Consortium work, which is really aimed at trying to create some really well-characterized whole human genomes that can be used to understand your analytical performance of sequencing. And so most of my contributions will be more on the end of analytical performance, but that tends to be an important aspect of sort of moving to the clinical clinic and understanding the first part of the process.
So, thanks everyone. Welcome here. Welcome to everyone else. I said when we had an email sort of thinking about what we were going to talk about today, my first question for integrating genomic and patient data, I said my first question was going to be, why haven't we solved this problem yet? What are the issues that are preventing us from solving it? But perhaps that was premature because perhaps we have to identify or at least define what the problem is. So maybe from all of your personal experiences, and I sort of asked this as well in the emails, what are some things you've struggled with? Maybe you could each just briefly speak to, in this area of integrating data, something you've struggled with and what you think the problem really is. And then we could talk about why we haven't solved it. So, I mean, in any order, we don't have to... Yeah, go ahead.
All right, so phenotyping. I know this is a genomics conference, and we've had like our fill of genomics and collecting a ton of data, a lot of base pairs being sequenced. But one of the biggest problems for us is linking that genotype to phenotype, and it's really around the clinical phenotype. So, for instance, my background is in oncology. And when we do clinical trials, we have clinical endpoints of partial response, stable disease, progressive disease, complete responders. But when you start to look at genotypes and you say, okay, well, this genotype is associated with, say, a stable disease. Stable disease is plus or minus like 10 to 15% tumor growth or tumor reduction. When you start thinking about that from a molecular phenotyping way, there's a lot of biology going in there that's not captured in the clinical data, in that phenotyping data. So to think about, and that's in oncology where we've been doing quote unquote precision medicine for decades. So when you get out to other diseases like rheumatoid arthritis, where it's a pain threshold, or mood disorders, where somebody's, how on a scale from 1 to 10, what's your depression like today? You can imagine trying to link genotypes to those phenotypes gets really, really difficult, if not impossible. And so my biggest problem is the standardization of phenotyping, clinical phenotyping, and how are we going to solve that problem?
Yeah. So, well, there are different waves of things we've been stuck to, and some of them we managed to solve, some of them we're still working on. One of the things we put a lot of effort at the beginning was answering the question, is next generation sequencing clinically ready? So can we have the reproducibility? Can we call reliably false negative and false positive? Can we actually make this technology ready to be clinically relevant? This took us a lot of effort, and Precision FDA is one of the main mission of it. We worked with the government. Is a fundamental question that we had to address, and we felt we reached an ability now, thanks also to the guys at Genomics Boston. We are proud users. So we had to sequence thousands of times the same NA121878 sample to actually reach a chemistry and ability and a pipeline that we believed was going to give us consistent result and high quality enough result. And this was the first kind of major effort that we had to go for it. The second that I see is the amount of data that we are collecting and the kind of research that we are doing. For the first time, at least is trying to merge patients and healthy people. So having statistical distribution between correct population and giving, can they hear me? I feel like, yeah, you can hear me. Okay, great. So this was the second part. Many of the results that we were seeing at the beginning were kind of biased either by the population at the cohort structure that has been built or addressing just specific kind of sick people. Simple signals like BRCA1 and BRCA2 signals, they are starting to have different weight once you add in the cohort of the study healthy people. So building a correct stratified statistical pool on which to run analytics, that was the second struggle. And the third one is for sure phenotypes. One of the, say the next conference next year, Phenotopics, could be an amazing title for a conference. In a way and in another, we managed to build a digitalize in a way and another the genome. There are many nuances, problems, we all know them, but still we haven't yet touched correctly how to onboard the stakeholders, hospital, doctors, to correctly do phenotyping at scale. We managed to do genotyping at scale. Phenotyping is still a wild, wild west that we all together have to understand how to map.
So one of the challenges I see from the technical side that's still really not solved is understanding how well you're doing for some of the more difficult types of variants and more difficult regions of the genome. And a lot of the genome we do pretty well at this point, I think, and the percentage is probably up for debate, but maybe somewhere between like 85 and 95% of the genome we do quite well at this point. But even understanding what that 85 to 95% is and which types of variants you're detecting well is still not really a solved problem. And so both understanding where you're doing well and then also understanding which types of variants you either are not doing well or you just don't know how well you're doing right now because we don't have good truth data or we don't have even examples of those variants in some genomes. And so sort of sorting out those issues and understanding, both getting better benchmark sets for more difficult regions and more difficult variants, and better tools to understand how well you're doing in different parts of the genome and for different types of variants is one of the holes that I see still existing.
Yeah, and to add to that, diversity. Diversity in the data sets that we use as references. I also would say privacy, being able to maintain the privacy of de-identifying the data. I think that, we at FDA are interested in safety, efficacy, and quality. So our focus has been on the analytical validity of the pipelines, and that's something that why we're bringing the community together. And in doing that, through challenges and through offering a lot of different carrots to people, like compute resources, competitions. We're offering a lot of things to people to become part of this community. But one of the another big challenge we have is getting people to share. Obviously, we appreciate the data that you've provided and many of the people that have provided their pipelines and participating in this community we're trying to establish so we can learn more and gather data, and people can speak to the experts and provide their expertise, and we can learn from each other. But it's difficult to do in sort of an area where there's a lot of confidentiality in not just the data itself, but there's corporate confidentiality, there's issues like that as well.
So thinking about the topics that you just brought up here, specifically the phenotype information, and then you mentioned the healthy versus sick, and the idea that we need healthy people. Do you guys think that it really is healthy versus sick? I mean, when you're thinking about common complex disease, does the line between healthy and sick get blurred into you just have aggregate phenotype information and then you see what happens? I mean, certainly we know for certain classes of disease, it's certainly sick, but when you're talking about heart disease or something, what do you think about the difference between healthy and sick there and just having good phenotype information?
That's the interesting question. And one of, let's say we call healthy people in a way that because we start the nucleus as a service where we actually sequence people that don't have any, let's say, obvious pathology yet in a way or another. So the line is not obviously well defined, and it's defined by the amount of phenotype that we can collect. But the effort of, and this is also some kind of a paradigmatic shift in how we manage health. We should have full genome sequence. It's, in my vision, a technology that is really groundbreaking. The moment is used across the border, no matter if I'm, let's say, diagnosed of a specific pathology or not. Because the signal that we can collect when we do it really at scale, then is the real signal that encloses the population structure. One of the main confounding factors every time you do any model is the population structure bias. And then there is the cohort structure, so what has been selected or not. Having an approach where you start to do really full statistical ensemble, so you have the complete population in a way and another, that's the definition of healthy or sick in a way is blurred over there. You start to have reliable signal, and you start to have real kind of p-values that you can start to use. And the other part is also important to understand where the technology, and coming back to the technical problems, where the technology can be used reliably or not. So when can I call or not? Examples are trinucleotide disease or repeat, let's say, insertions, which are typically one of the critical problems when you do NGS. Can we actually, does it make sense to build phenotypes, or can we call reliably? Okay, so even when you build this kind of signal and databases, there is a second filter that what the technology can actually predict and can read inside the genome. And once you pass that filter, you can start safely to build a signal across the phenotypes. If you hope to build a signal on those two databases, phenotype and genotype, on a piece where the technology is not performing well enough, you're just producing noise most of the time, or risky signal in a way. So it's a stratification of having the correct data ensemble that really represent the population that you want to address, filtering over the capability of the technology. Once you get that verified, vetted data, that you really are secure and safe that that's the read, then I can start to do models over a good phenotype.
One of the things you touched on too was the whole aspect of the clinician and trying. Yeah, I thought that was really interesting because they're going to struggle with this. They're not necessarily trained.
Yeah.
They're going to have to interpret this data. It's going to if we have that fully integrated data, they're now going to be at the point where they have to have some way of understanding that data. We, of course, at FDA are going to have to figure out how to help with that. But you touched on that, but what are your thoughts related to that?
So when you buy and you acquire or you touch different cohorts done by different clinical teams or done in different hospitals, you understand the absolute non-uniformity of the phenotype calling. So when you start to acquire and see databases that have been done by different countries or different doctors, probably in the same hospital, the way they tag the phenotype is sometimes fundamentally different. There are people that are very experienced, they use OMIM codes, they get down to the very defined leaves, you have very precise database, you have entire cohorts that lasted for 20 years, where you have a piece of paper and the kind of, let's say, human written disease or pathology by the doctor. This is a significant problem because there are 20 years of experience that are very hard to capitalize in a data-driven approach. So the way we need to work, in my idea, is helping the stakeholders and the people in the clinical world to understand how they can produce phenotypic data ready to be used in a data-driven, let's say, healthcare system and clinical system. Because by the amount of the effort and the time and the money that are being spent, the differential amount of knowledge and education at the beginning to know how to correctly map a phenotype give an exponential, let's say, added value on the pipeline, because you have uniform, or in a way at least reliable phenotype that is tagged with an international standard. Standards are there. It's not that they don't exist. FDA made an amazing effort. Lots of people made an amazing effort. I believe it's just an effort of knowledge and education on the very beginning of the clinical practice, how to tag correctly and how to intake digital information.
So I maybe want to throw a wrench in that, or at least ask the question that, Justin, like a lot of the work that you've done and we've done on the genotype and genomics side, the idea is to achieve data harmonization in a preventative manner where we go and we figure out best practices, we validate pipelines. That way, once they're run, the data are harmonized. I'm wondering, for phenotype information or clinical information, is that realistically possible with all the different sources, or should we be looking for more a corrective way of harmonizing the data? Regardless of what the data we look at, we get in, we're finding ways to figure out how to harmonize that after it's been collected. Or is the only way to go in a preventative way where we're expecting people to follow some set of rules up front? Is that, we know it's feasible for genotype and genomic information, is that really reasonable to expect for phenotype or clinical information?
It's gotta be. I know. I mean, so the way I look at this is, there's two ways. I like what Ricardo was saying about the education of the next generation of clinicians. They're gonna have to know genomic medicine. They're gonna be forced to know it. They're gonna either get on board or get off, right? And that's gonna be for community physicians and everything. It's gonna be part, I mean, it already is, part of their training, and you start to see this paradigm shift as far as they know they're digging into the science. They want to do the phenotyping in a standardized way. They've been hearing this. The other thing is, so how do we deal with this now? I was mentioning this today at a lunch panel also, where maybe we start small. Maybe we start with, say, a clinical community. Let's take Cambridge, for instance, right? Or a small, or like an academic center, and many academic centers are starting to do this, right? In certain disease areas where they're like, we're going to genotype everybody, but all of the clinicians, all the physicians are going to be trained on how to have a standardized phenotyping and standardized language use. And you're starting small, and then you say, okay, are we getting anything out of this? Can we link genotypes to phenotypes to the point where we can make clinically actionable decisions? But I think there's got to be kind of this proof of concept of doing it right. And I don't think that's, I mean, even in oncology, where we do it a lot, I don't think it's perfect at this point. And it may never be, but we have to try it, we have to attempt.
So you don't think there's a way to figure out after the fact?
Right now, I mean, okay, take ICD-9 codes, for instance, in the States, right? Those are billing codes, right? So they're about billing. They're about getting reimbursed. Many of those, may not be accurate, right? It's about reimbursement. So, I think there's a way. I mean, you could do like text mining. You could do some of these things where you're saying, okay, has the patient been treated on these certain medications over, longitudinally over, say, 6 months or a year? Can you somehow derive a correct phenotype from all of that type of data? Possibly. I don't know. But that could be something to
And I guess with the people on this panel right now, one, Elaine, with the Precision FDA, you've brought up the point about like the competitions, right? Putting the challenges out there. And it seems like these types of problems, hey, here's a bunch of data, can you find a way to harmonize that? Maybe there's some sort of crowdsourcing there. And then also maybe some of the machine learning, Ricardo, this is your background, right? Is there an opportunity to use more advanced technology to actually harmonize data that's very, heterogeneous to begin with, as opposed to trying to expect them? I'm just harping on this because I feel like if we're expecting to go out and train all the clinicians to behave a certain way, good luck. I'm going to go do.
Quality of the data is a really big issue.
Yeah.
I think that's something. I mean, we can harmonize, we can do lots of things with data, but if we have really poor quality data, we're going to have a lot of problems. And because we're so decentralized in the way that we do health in this country, it is a big issue. I don't know how to specifically solve that other than talking about standards and, I think providing more in the way of clinical pathways and things like that, really providing more information to make things easier for the clinicians, more, standard ways of handling certain information. And I think that's going to be a lot of it, and doing much more detailed analysis. But yeah, quality of the data and actually having people understand the data, looking at the data and identifying the data is important as well. Because, when you have a clinician who's, pressed for time and filling out, a health record, I don't know how much you're really going to get. And then you're expecting them to have even more information to look at and analyze. We're going to have to provide ways to analyze that data for them as much as we can.
So Ricardo and I were having a conversation earlier about this also, but there's different ways of phenotyping, not just clinicians phenotyping patients. There's also self-phenotyping. There's, the use of wearable technologies and cell phones and things like that, but there's also caregiver phenotyping. It's something that pharma is exploring for, say like Alzheimer's and dementia and some of these other diseases where, you all know, like with your spouse or your kids or something like that, if something's slightly off, you know it. You know it better than anyone. And so I think there's, thinking outside the box a little bit more as far as, how we're doing the phenotyping, may not just let it in the clinician's hands, so to speak, is another thought.
So just to give you an example on the machine learning side, let's say most of you know the various neural networks and image recognition systems that are coming out. So it means that there's an algorithm that look at a picture and tells you, let's say if there's a bird or not, if there's a cake in the picture, anything that, let's say object recognition. Now these databases I've been trained with typically triple check construction. So the image is exposed to a pool of 3 people, and 3 people are asked, is there a cake inside or not? Is there a bird inside or not? And you accept the answer, maybe only if all the people agree that there's a cake inside, okay? That's how you build. Amazon Mechanical Turk is a typical infrastructure where you streamline millions of images and you collect millions of opinions and you require strict, let's say, truth about all the observers. Now, if this is the amount of requirements that we build to classify if a picture as an object or not, nothing less than that should be required for health information. So we need very reliable data, and we need to find a way to build this highly reliable data, because every single other industry that is doing at scale machine learning and data-driven strategies require this amount of precision to train their models. And so this is a standard in a way that we nailed in different industries. Healthcare is way more complicated, but this is the goal we have to head to, and I'm sure we're going to get there.
And we're trying to do that on a small scale with Precision FDA, with the crowdsourcing aspect of it. One of our biggest challenges, of course, is engagement, and getting people to be engaged and stay engaged and have conversations like this in that forum. And we're looking for leaders in the community because our goal is to get to a self-sustaining, largely self-sustaining community. So, and we can address other topics other than what we're addressing right now, other than those things that are specifically interested in, related to analytical validity, for example. We're probably going to, our next challenge, which will issue maybe in the August-September timeframe, will probably be related to cancer or something like that, and hopefully that will gather more information. But we certainly will accept challenge ideas, discussion topics, things like that from the community, because we do believe that that's how we're going, the more data, the more people participate, the more information we have, the more we'll learn, as you say. And so this is a community for you, this is a community for you to participate in, and we would like to see you taking advantage of that and the experts that are available on that community.
And Justin, you had brought something up in a conversation or in an email, finding a way to visualize appropriately certain comparisons. And I think this idea of visualization and, like you said, getting people to buy back in, where if you're a caregiver, if you're an individual thinking about yourself, if you're a clinician, if the idea that you providing information in an accurate way that abides to certain standards, if you get something back, if you have that investment. So this idea of visualizing these results appropriately is important. I always think about how active people on Facebook with kids are about posting, oh, my kid is sick, here's what happened, here's the entire story, I'm clicking through, I don't care. Other parents care about what happened, and that kind of feedback and community engagement, we all say it's a great idea, but then you get into issues where you're limited by privacy concerns on what really could be effectively shared. So Justin, maybe could you, and I know that you're on the technical end, but the technical end isn't encumbered by a lot of the privacy issues that we face moving forward. So on your end, what have you found ways to engage the community and visualize your results? What are the lessons you took away from there?
Yeah, that's a good question. So, so one of the newer things that we've been talking about related to this is, so a way that we've sort of related to this idea of having multiple people look at the same picture and decide what's true is that one of the things we found really useful in determining what's actually a true variant or not is looking at the data from multiple data sets and seeing whether we see any evidence of bias or if we think this is a true thing or not. So one of the things we've been thinking about is, can we make some sort of a platform that would make it easy for lots of people to look at different sites in the genome where we're not sure what the correct answer is, but it would be useful to have people manually look at them and say what they think the truth is, and probably have multiple people look at the same site because any individual is not perfect. And maybe I'm a little skeptical as to whether non-experts can do this in a robust way, but I think for at least a subset of the genome, probably even non-experts can do it. And so it occurred to me when you were talking about the phenotyping or electronic medical records or interpreting that whether something similar could be done for that, where you have like a clinical report that you ask people to interpret or highlight what they think the disease is, and maybe initially do it with experts, but maybe non-experts could do it in some cases also. But actually, like, getting people to do that is something that we haven't quite got into yet.
Sounds like a competition.
Whether it could be framed as a competition, I'm not sure. It probably could be, but yeah, I think that it could be a useful way to engage the community, maybe.
Great. I think we're at the halfway point, and while we all think about how we're going to answer these next questions, I hopefully can open up and have you guys ask us questions or make points. You can come pontificate like we've been doing. It's fine. But definitely engage, and then we'll loop back and see if we could sum up a couple of points. So any questions for any of the panelists or new ideas? Things you're interested in hearing about?
Movie reviews.
Yeah. We got a runner.
Can we ask questions to the audience?
We got
Yes.
a runner. We should all cheer. Make them jump.
But yes, we'll do that next.
So we talk about the standardization and getting good data quality, but one of the missing points that we have is when you're located in a place like Boston, you have clinical oncologists that are specializing in individual type of, but you're individually going into each of those type of subtypes of cancer. How do you guys see getting the knowledge base of interpretation of those results to the community hospitals that don't have those access and maybe only have a surgeon that's going in, and what they're going to do is they're going to cut. They don't know precision medicine. How do you bridge that between the cutting edge and the bleeding edge science and genomics and what's being done in the rest of the country? Because right now they're estimating about 50% of all patients get a tumor or get some type of oncologist to look at them. That means 85% of all United States is not getting that level of service.
Yep.
Sorry.
No.
Nobody else would probably do it, but if
Someone want to summarize the question?
This was more about outside of big academic centers where there's a lot of specialty. There's a lot of people that have look at omics data or think about precision medicine and things like that. Moving that to the community-based care and how to educate. As we were talking about before, how to educate that community on the relevance of this data or even the actionability of the data. Coming from pharma, I mean, obviously we have to, on our labels, if there is a biomarker or a patient population that is going to respond to a certain, that has to be on the label. That's kind of a no-brainer. If you want to give them this medicine, they have to have this test and they have to have X results. As we start getting into the multi-omics era and the large data sets and making clinical decisions off of that, I think that's pretty much what the question was more about, right?
About accessibility of the knowledge and to community hospitals and people like that. I think we're hoping, our plan is for Precision FDA to be something that has a diverse population, including clinicians and people like that, that eventually it will expand to that point where it's offering information and expertise that can be utilized to anyone. So we hope that it will cross those barriers, and I think that's definitely an intention we have. How far it goes and where it leads will definitely depend upon the community that is participating. So, but we have big hopes that it will remove some of those barriers.
So I have a big hope on the patient side. I tell you an example. Now, if you go on the homepage of FDA, there is a beautiful page of pharmacogenomic effects. Okay, there are famous kind of mutations that interact with their metabolizing or some specific problem with the binding receptors, and there are drugs with respect to a specific mutation that might actually not work at all on you. Okay, it's something that you don't know. An example, typical, the top I'll take is statins. Okay, statins have a very strong, let's say, signal on specific mutations. Now, when this simple concept is explained to anyone, okay, let's take the most wonderful case and powerful advocate of health, a mom, okay, and you explain about the potential risk of a specific mutation that or not with respect to pharmaceuticals, they immediately understand the value of having qualified and quantified reports on their genomic profile in one shot, okay? So if we can explain and we can make the case that the health that you're going to receive, the service that you're going to receive, is going to be better if you expect and force and ask a correct analysis, digital report, e-report, correct qualification. There are an entire bunch of patients that meet online to find second opinion and so on and so forth. This is something that is happening under the hood in a way or another, but if we leverage those kind of interests and those stakeholders and explain that if they help and if they push to their own clinic to get upgraded, to have quantified information, to have digital reports, they are the best advocates. As much as they are the best advocates, once the pharmacogenomic profile, they get to know it and so on and so forth. So one of the big efforts that we do as well is explaining the final user, everyone, that anything that is digitalized on your health gives a much better value for the ecosystem, but for you at the first point. So I have a big kind of hope on the patient effect.
Yeah, my sense is that as algorithms get better and better, that it'll be easier and easier for it to spread to more places also. Like on the genome analysis side, it only probably a few people could do it like 10 years ago or so, and now like more and more people are able to do it because the tools are becoming more streamlined, and so hopefully that will happen also on the interpretation side of things, that it will be computationally easier to explain it to anyone. But
I think you're absolutely right, and I think the other thing that'll drive that is, again, goes back to actionable data, right? Where we have a plethora of therapies to offer patients with certain, genetic abnormalities or, mutations within their tumors or things like that. Like I said, we've been sequencing tumors for decades almost. I'm old. And so, but, think about the amount of drugs that are actually out there. We know so much about the cancer genome, about what drives tumor types and things like that, but there's only a small handful of drugs that are actually select this patient and they have a great chance of responding on those labels. So I think the more drugs we have in that pipeline to use in a genomic medicine type of framework is going to really push that understanding, even a community clinics and things like that. I think right now there's probably a lot out there, like, why would I sequence this person's genome if I can't do anything about it? And I think we have to get past that type of attitude. Right?
So I'm not sure, I don't know, but if you are anyone from a patient to a caregiver to a clinician, is there a web MD, PhD resource you can go to where I have some sort of, more than just like this part of my body hurts, but like I have some phenotype information, maybe I have some genetic and genomic information, where I go and I have that conversation? Is that what's being built at Precision FDA? Is that something that already exists? Because this gets to the point of, if I'm the only doctor in rural Pennsylvania and I have this information, I know I'm not necessarily an expert, where do I go to even know if it's meeting the existing standards and maybe get some help interpreting it, whether that help be other individuals or tools and resources that you're pressing buttons on?
Right. So if the analytical software that we have today that analyzes the data that's coming out from the sequence data, the FASTQ, if we're not even sure what's good and what's bad, which is what we're trying to figure out now, is how do we know that your pipeline is good and their pipeline's not good? We have to figure that out so that we can say to somebody, this is, a good pipeline. It'll be able to, find the noise. It'll be able to, target the different areas. It'll be able to understand what's in the genome. How can we can't get there. So we have to get past that hurdle first. And I think we're making a lot of progress. One of the things that I think we discovered in the first challenge was the pipelines that were there were really good. Actually, we were surprising how good they were in finding the variants, et cetera, the known, variants, the truth. And we're trying other aspects of that. Justin's challenge, for example, the truth challenge. But that's the first step for FDA, is really getting to that point. So would I think that at some point somebody would want to go up on Precision FDA and, actually be able to run their own, genome through their own file through a pipeline? I would hope so. I would hope we would get to that point. I don't think we're there there yet. They would be able to have that tool.
Right.
There are tools there available to them to do that, but I don't think they could at this point. I think you need to be a bioinformatician to really be able
I guess
to do that.
because I have been able to see and play around with a bit Precision FDA, even though I wouldn't probably not be a typical user, but I have seen it. And there's some really nice like community forums and message
Yes.
boards and the ability to share data. So I'm wondering, in that interim, while we're getting to that point, is Precision FDA a place where if you have some data and maybe the pipeline to run actual algorithmic analysis isn't there, it's a place where you can go ask other people, the appropriate forum.
I think it's a great forum for people, anybody, to go and talk to experts, because this is where the experts are coming together, or that's what we're hoping they're doing. And, it is an opportunity to do that. And I think also there are tools, there are pipelines that are available for people to use, not just the discussion forum. And there's a lot of data there today. There's, we have a lot of reference data. We're hoping to get more diverse data. We're hoping that, the million-person cohort, as long as it's de-identified data, we can't use data that's not de-identified and doesn't have consent forms associated with it. But yeah, I think it's a great place for people to go to, not just the scientists to sort of learn.
I guess since we're being filmed, I don't know how many cable stations it's being broadcast on right now, but because I'm not sure, anyone can sign up for Precision FDA? Do you want to walk through how that actually works? If people here, experts, wanted to go and be experts on Precision FDA,
So
how that would work?
There's basically two kinds of access. There's browse access, which is, you have to remember, this is a beta. We're trying to see, is this something that's really going to take hold and it's really going to happen? We really want it to. So there's browse access. You can request browse access. You'll get it immediately. And you can see anything that is on Precision FDA.
The forums?
You can use the forum. If you want to collaborate and be a collaborator of Precision FDA and actually, add data, pull data, add tools, pull tools, participate in competitions, all you have to do is when you request browse access, it will tell you how to request collaborator access. That comes to me or somebody who I work with, and we look at it. We may call you. And really, the reason we set that up is because we want to encourage people and we want to develop relationships with people in the group. We hope that at some, it'll get so big we can't do that anymore. But right now, that's a big part of what we're doing is try to engage people, make sure they have somebody they can talk to, and also make sure that they're not somebody who's just trying to, hack our system or something like that. We want to really get to know the people that are part of the community.
That's great. Ricardo, you had a great idea of giving us an opportunity to ask questions too. And I think this is a nice punctuation
Every
point.
time I do a talk, I always ask the audience how many people have their full genome sequenced, or at least a genomic test. And this is the best crowd ever because we are all professionals in the field. How many of you have a full genome sequenced? Or full exome at least? Okay. True, I do have. Okay, so 6 people in the room, 7. Okay, 7 people. And so I understand, and I'm sure that all of you believe that this is an interesting asset for your health in the long term. Okay, so and as long as a cohort of people that work in the field don't see or don't act on the value of getting sequenced, and all of you know someone that can sequence you for sure. If you don't, I'm happy to give you my address. And if us, our cohort of people in the field, they're not still actively building information on their own health, then we are still far away where the moment that any patient and anyone in the United States and in the world will actually be responsible of their own quantification of health. Okay, so and ask yourself why I haven't done it yet. So you all have access to sequencing platforms. You maybe work with a sequencing platform in your lab, and you still haven't done it. I mean, there are several reasons, and I love to know all of them, because maybe we can go through and try to check what is.
Yeah, one of the reasons why I haven't done it yet, I've had some 23andMe, and then I've had a pharmacogenomic genes tested, and I would get my genome sequenced. I wouldn't not do it, but I just think a lot of the information that we think we can get from a genome today isn't really. It can be misleading, or it may not be useful right now, especially some of this risk modeling for common diseases. And so I think it's a little bit. We have to be careful in what we draw from a genome today, because we know we're going to get a lot better at that. But I do believe that what we can get out of a pharmacogenomic profile is useful today. And so I got that done with someone I know who runs a company in Australia who does pharmacogenomic testing. And then I was in this situation where we don't have a federated electronic health system which I can deposit that into. So if I walked into a hospital, unless I had my report there with me or some sort of digital copy of it, then I would have to otherwise contact every single hospital or every single doctor to give them that report. So there's practical challenges in disseminating the genomic information to the health system so that if you do get sick or have an emergency, that they actually will have that information readily available.
So those are great points, and I think, it's like anything else. It's like, well, if I look, I'm going to find something, and am I going to find something bad? So I don't want to look. An example I used earlier was colonoscopies. It's amazing how many men and women don't get colonoscopies. Colon cancer is very preventable. It's very curable if everybody had a colonoscopy. Now, some get missed. I know, like, even if you have a colonoscopy. But the uptake of colonoscopies is atrocious. And so, but when you talk to people, it's like, well, there's the prep. But, I don't want them to find something. What if they find something? How am I going to deal with that psychologically? If we've got predictions for, like, Alzheimer's, I don't want to know. I don't think I want to know right now, because, like, if I forget my keys, I'll be like, uh-oh, here it's coming. Great, right? And because there's nothing I can do about it, right? There was a patient where we were doing circulating DNA, and we found that, we could monitor the regrowth of their cancers by circulating DNA p53 mutations, right? So do we really want to do that? Do we want to tell the patient before there's any imaging or any type of phenotype or any type of morbidity associated with it that your cancer is back? What does that do to their current life? What does that do to their family? So there's a lot of these questions, I think, that go into, do we want to know? Do we want to look?
Because I've been hearing,
I follow up on
okay, go ahead.
real quick. So I love your point. And I tell you my personal experience. I got my genome sequence, and at the beginning I did by myself. I had the file. So I started to join against every single database I knew. CleanVar, DBSMP, and so on. Okay, after 2 hours, I wanted to buy a ticket for Indonesia, surf my last day of life, and just let myself die in the ocean. Because I thought I was going to die of everything. Or literally everything. I had every single pathology on planet Earth in there. So that is a problem of phenotyping. So we have databases that are not very well cleaned. And that is a second problem, is that this kind of information, in our experience, should be managed in a health process. So you need a professional to read that amount of data, even if you yourself are a scientist, okay? You need a doctor that actually do some differential diagnosis and probabilistic calling, for which, okay, yeah, you have a SNP that in 6% of the population is correlated on something terrible, but there is no family history, there is no, and so on and so forth. You start to screen it down and you apply some basic kind of medical diagnosis process where you hit only something that is reliable. So in my understanding, we need to integrate the doctors in the process. I see it as a kind of a counterproductive process, reading your own information, unless you have experience training in the medical domain. And that's why we kept doctors in the loop, is a fundamental part. And second, we need better databases. Because if we don't have better annotated databases and phenotypes, professionals in the field will look at genomic sequencing as something not reliable. And if we ourselves don't trust the database that we use, we can't expect to push them to the population, right? So we need to at least find a procedure that makes us all confident, yes, I want to get sequenced and I want to get this procedure done, and this data join done, because I know this is really informative. So I feel your pain, and I felt it on my skin. Then I go to the genomic teams, they explain that I'm not dying, I'm still here talking to you, but still I had a lot of actionable information on my personal health that was vetted at that point, so I could take care of only a few things.
Well, I just wanted to add that according to my insurance company, my doctor only has 15 minutes with me. So we definitely need tools to support them, and risk models, things like that. So we can't depend completely on them. We need databases of information and tools to analyze the data.
I guess, because we've been asking this question for a long time, like, should patients have their own information? What would they do with it? Is it actionable? Is the data correct? Is it useful? I have a gym membership I never use. I spend money on things and I never go for my health, but I just spend it because it makes me feel better. Whether it's actually useful might not be relevant. My question is, how much would a genome have to cost where people would just do it and not really care? If a genome cost $1, if getting your genome sequence was $1, would everyone who's here like, I don't know if I want to know, would they be like, all right, for $1, I'll just have it? So is it really, how much does it come down to cost, or how much is it actually these concerns on actionability?
I don't know, and it's a great point, because it's like, insurance. So many insurances in the States will cover screenings, right? Mammographies, colonoscopies, and stuff like that. There's no cost to those things. So I think, and again, I mean, these are things that are severely actionable, that can lead to a long, healthy life, and people still aren't doing it. Again, I don't know what the answer is, but they're still not taking advantage. Simplicity could be something, is it? But I don't know. I don't know the answer.
It's either, are people scared or are they cheap? And you seem to be arguing that they're scared.
Well,
that's why you don't get a colonoscopy.
Well, I mean, because if you're insured, like I said, it's paid for. I mean, so we work really hard, because to intercept a disease, the diagnostic, especially in oncology, is huge for us, right? Because so much of it is curable at those early stages. And so, we're putting a lot of effort into that, and into making it easier. I know, like with circulating DNA and these DNA stool tests and things like that for colorectal. But we still are concerned with what is how are we going to educate people, and what is the uptake of that going to be? We want to get rid of the preps. We want to get rid of anything, clear that. But it'll be interesting to see what the uptake of that really is. How many people actually come in and get it done?
Yeah, I just got my genome sequence results recently, and it didn't really strike me until after I got them that pretty much any news that I was going to get was probably going to be bad from if there was any news that came out of it at all, and it's even worse if the news isn't correct. That it's,
oh, great.
But
Well, there's no great VAR where it's just like, here are all the mutations that make you a better person. Like, we don't collect data on health. We don't collect data on healthy people like that. We only have pathogenic type stuff.
And of course it isn't really bad. If it's actually actionable, then it actually probably is good in the long term. But even if it's actionable, it's sort of bad news initially that you have this disease. And
There is one point about the gym. Then I'll so there is one time in your whole gym experience every year that have a peak of presence, and I'm sure it does. It's springtime, okay? You're cool for the exam of summer, and then you're going to be more exposed, and your body's going to be more exposed, so you feel under exam, and everyone starts to run on the treadmill for just those 7 days to try to hit it. And this is the problem of I think one of the issues of genomic testing as well. So we don't believe it's valuable unless we are under a health condition, okay? And this is wrong in a way or another. So the best way to start to think about it is what is the best procedure that allow me not to get sick, or that allow me to take best practices as early as possible not to get sick. And this is the shift between merging the term health and disease and starting instead of actually practicing health. So having all the information that I can have to prevent getting sick, or to, let's say, having and practicing as gym as possible to get in somewhere, but not that you're not going to succeed, but having the best strategy early.
So, right. So basically some good news with your bad news, right? If you exercise and you watch your fat content or whatever, then hey, you're gonna be fine. You reduce your risk significantly. So some good news with your bad news. But also, they have to be accurate. Within some probability, they have to be accurate. Otherwise, insurance companies will not cover it. And even if they don't cover it, I'm not going to waste my money on it. And I think a lot of people feel that way, right? If it's not accurate.
Well, I mean, infomercials make money. Like people, that's always my big concern is that there's a huge opportunity for snake oil salesmen when you have these results, if they're inaccurate, like, oh, well, there's this paper that was done and we can help you because you have this mutation. For me, that's the accuracy, because I have people I know who will just be suckered into all kinds of horrible products based on their inaccurate report.
Right, and you're just making the case for FDA's role.
Absolutely.
Because that's a key role.
Hi. So my question comes with this whole, if you do get yourself sequenced and you're comparing it to like a colonoscopy, which a healthy person with no history of colon cancer is going to get once every 10 years, will you resequence or reanalyze the data? Because these variants of unknown significance in 15 years could become significant known variants. And I think our hesitation to call things wild type eventually, I think, will play into our favor, because better safe than sorry. But because that would actually motivate me to get sequenced. If fine, you don't know what's wrong with me now, or like I'm fine now, but when I'm not, you have that and you'd be able to reanalyze. So how would that be integrated?
So real quick, I think the analysis of the existing data, variant analysis is computationally cheap. You could be, I think, continual reanalysis of variant data, like week, month, whatever you want to be. But the point you brought about resequencing every so often, I think, is interesting. Whether you need to collect like every 10 years or 5 years, you go and resequencing. And that's not something, when we talk about sequencing tens or 20 of thousands of genomes, we're taking that snapshot and generalizing that. So I think the idea of getting sequenced multiple times is really interesting here.
So I think that's interesting. I mean, I've always come at it, from my training was in epigenetics, right? So we know that epigenetics changes as people age, in PBMCs, in gut epithelial and things like that, how that relates to disease and things like that. So the re-epigenotyping, so to speak, longitudinally, could make sense. I'm not sure about the genome. I think that's more of a technology. How is the technology better in 10 years at identifying variants? But also it comes back to the interpretation of the variants. As you know, there's so many variants in non-coding sequences that are going to be really important for gene regulatory networks and things like that. And that's going to be very hard to decipher, and people are deciphering it, and we're going to have more knowledge as it goes on. So knowing, it goes back to whole genome, versus whole exome, let's say. Whole genome is going to be this wealth of information that we are going to be identifying things in the future that may not be relevant, or we don't have the information or the phenotypic link now, but we could have in 10 years.
Yeah, I mean, the upgrade of the information is going to be constant. And the moment let's say, you manage and we find a way to correctly, in an anonymous way, share the DNA, the amount of information that you give helps to explain other variants and other signals that we didn't. But the resequencing, it really depends, I guess, on the state and maybe the pathology that we're discussing. There are tremendous advancement on cancer vaccine or cancer sequencing, actually, not anymore sequencing your germline in a way, but sequencing your cancer. And so you might probably get exposed to sequencing several times in your life for probably different tissues or different times. But the upgrade of the information is going to be constant for sure.
So I find it kind of frightening that the precision FDA thing actually has to have a reproducibility challenge. This seems to me like kind of a basic science thing that shouldn't have to get all the way to the clinical side before people are pursuing it. And you mentioned ClinVar, there's a huge number of conflicting reports in there where one person says pathological, someone says not. Why isn't that more of a focus, and what can be done to improve reproducibility?
Yeah, well, so there's reproducibility in terms of like if you're a single lab that's sequencing the same sample multiple times, do you get the same answer? Or analyzing the same data multiple times, do you get the same answer? And those are clearly useful things to look at it as part of your process. And at least in looking at data sets that are out there, it seems like the reproducibility within a lab tends to be much better than the reproducibility between labs or between different assays or different bioinformatics analysis methods. And there are a lot of reasons for that. And I think the reproducibility inside the regions of the genome that are relatively easy to assay is actually quite good. It's mostly in these sort of edge cases that are in repetitive regions or low covered regions or some of the harder parts where this reproducibility starts to break down. And so I think basically the first step is sort of understanding where we are reproducible and where we're not. And that's something that, probably should have been worked out a while ago, but these methods are changing so rapidly that it's hard to work that out at any time point. And so having challenges, I think, is still sort of a necessary part of the process.
But I think what he was talking about is a bigger problem of variant annotation and assigning pathogenicity to mutations, which is like a huge elephant in the room.
Right.
Of
Well, that's
the whole
definitely an
field.
issue.
But it's phenotyping, and I personally have, let's say, not a clear path on how to get there. What we knew is that we collected our own phenotypes. We built the entire pipeline end to end, and so we have phenotype analysis, high quality measurement, different machines for every single patient that comes in, full genotyping, metabolites. We did everything end to end, and only doing everything end to end, we started to understand how and the importance of the value of having high quality phenotypes. When you have ClinVar is a great effort, but unfortunately, as I said, many committers, they are highly, let's say, conflictual in many ways. How to make a sense of it, that's, I believe, what FDA is doing. So is building a platform where it clearly will move slower than the amount of results that we are producing out of the labs, because you need to build coordinate effort and to vet every single step. But I believe is the good way to go. And once you pass some of the qualified tests, you can say this is something that has clinical relevance for real. So is FDA approved, we should go. It can still be an error in a way or a partial result, but this is the best way we got. And for the calling, I mean, I'm sure you're comfortable with all the variant calling procedure and so on and so forth. When you use different software right now or different bioinformatic pipelines, the ability to define if something is a mutation or not is still not yet there. There are some software that say this is a variant, this is an indel, this is not, then you have to phase and so on and so forth. The fact that FDA put a gate on that and say whatever you do, you need to respect some minimum requirements that we believe are shared across all the industry. Those are the ways you can start to pass and have a platform where in those things we didn't fail. Here we got good. Here we can't say. And there are many things that we still can't say with NGS because we are finding the way to do it programmatically and reliably. Repetitions is a typical problem. There are some pipelines that find great repetition signals. Huntington is a typical disease, trinucleotide repetition elongation. You go over 60, you do short reads, 150 base pair. Some pipeline call it, some post-processing call it, some pipeline don't call it. And this is still under development.
So what you described previously, which would be a deep phenotyping effort to categorize both healthy and unwell people and have their genome sequence, it sounds like something that the community. It would be a huge resource for the community. But presumably that information is never going to be available to the public to access. So can you maybe comment on that? So I'm also involved in a study in Australia with deep longitudinal phenotyping of healthy and unwell people, but I think there's a massive sampling bias in genetics that exists towards diseased individuals, and we need a stronger negative control group. But yeah, if you could comment on the whole data sharing and how the value of something like that for the community that's generated in the private sector, what's the position and role of that going forward?
Okay. So we are collaborating on many different projects. We collaborate at Precision FDA. We shared lots of data with Precision FDA exactly because we knew that is important for the ecosystem and so on and so forth. The way to finding the right way to share data has one fundamental issue of anonymity. So it's not any data set. So when you share this kind of data, we still don't have the technology and the protocols to be able to share, let's say, genome information without breaking the anonymity of the patient. And we recently worked on a paper that is going to be out soon about re-identification through genome sequencing. So there is an inherent kind of building block that we need to build to ensure kind of data sharing without breaking the privacy. And this is one of the factors that I believe is more important. On the strategy of sequencing healthy people, I can't agree more. When you have a statistical distribution that is complete, some of the signal that you find in literature, some of the p-value, they either disappear or they get completely different. And this is something that we got through experience. I think we are getting better and better, and the ecosystem is getting better and better. But the sharing thing, I think as well is an interesting business opportunity. So if anyone can find a way to ensure double-blind sharing with cryptography, maybe Confluent or any kind of approach that preserve anonymity, maintaining the information of the data, it's a holy grail. That's what we need.
So I'm not from the field at all. I'm here as an interested consumer. So I'm sort of your, the patient that you keep talking about, right? Or I'm a healthy individual interested in the whole idea of genome testing and so on. So how do I get my whole genome sequenced? I have no friends who can do it for me. Will you do it for me? Second, if I'm interested in sort of giving away the genome data to research purposes, what avenues do I have? And I would be interested in finding out rather than not finding out anything that could be found out. So for people to get involved in this, is there a forum? I'd also ask is there like a resource of experts? So, if in 10 years in the future I got diagnosed with something, and I want to know who knows how to interpret this stuff correctly with all the uncertainty you have in phenotyping and everything, where is the definitive list of people who know anything about the subject for a consumer?
We should have had you lead the panel.
So to answer one part of the question, if you are interested in sharing your data publicly and so that anyone can use it, the Personal Genome Project is a good project to join. And if you search for, I think it's personalgenomes.org, that's actually how I got my genome sequenced and shared it.
Do they still sponsor it?
So they will sometimes sequence your genome for free for you, but not always. But if you do get your genome sequenced outside of that, you can contribute it there also. There are other resources. There are ways to pay for your genome to be sequenced now, and I don't remember the names of them now. There are a couple that are done in association with Personal Genome Project, or at least connected to it in some way, where it's like $1,000-ish, I think, to get it sequenced. You can also join different studies. HLI probably is one option also, at least.
Yeah. I mean, we have this service. More than happy to invite you down in San Diego. And what we do, so one thing is getting your genome sequence, which, just to be clear, we are talking about a file. Okay, so when you will go around the venue, is a BAM file or a VCF. So it's a list of the mutation that comes through a machine. An analytics part, that's the bioinformatic pipeline that we discussed, okay? And you get your mutations in a way, respect to a reference, whatever it is. Now, this file needs to be interpreted. And the interpretation is what we're discussing with this gentleman. So you need to choose, is this interesting, is this not? Is this a risk factor, is this not, okay? So joining this amount of information, doing by yourself is a daunting task, and also kind of goes, that's the risky part of doing by yourself, not having a clinician and a genetist. So the service and what I guess is more important for you is not just getting sequenced, but having a report that gives you more information than what you had before, okay? And that is what HLI is doing. There are other competitors that, I mean, we can chat, more than happy again to invite you. And this, I think, is the differentiating value. So sequencing will become a commodity fast. So we will get to almost $100 cost in a few years for full genome sequence. The added value, so how to empower your best practices after that, the report that you receive, that's what is really the value that you want, okay? So your genomic profiling in a way. But more than happy to invite you now.
What about Precision FDA? Browse it? For someone...
Oh, well, yeah. For access to experts, you certainly could go and ask questions in the discussion forum. It'd be interesting to start having patients involved, for sure, because that's a place where you can find many of these people who know a lot about the area. What the focus is for Precision FDA really is right now is this a good pipeline? Is this do what it needs to do? Does it find the variants it needs to find? That kind of thing. But it will evolve beyond that. So
how does this work in a clinical setting? So if somebody with cancer comes to their oncologist, how do they know that that person knows how to interpret the genome data or not? Is it just a crapshoot, or based on where you go, or do you just go to one of the best hospitals and hope that they know what they're doing?
Oh, for oncology, it's unique, right? Because cancer genetics is a lot different than your germline genetics, right? So making clinical decisions comes from the sequencing of the actual tumor tissue, okay? But if it's, say it's another disease or another disorder or something like that, that you've had your genome sequenced, I mean, you're going to have access to that. You will be, if you go to a community hospital, are they going to have the expertise to act upon or decipher some of this? Right now, probably not, as we've discussed. So you are going to have to seek out experts, and this is where Precision FDA can really help with some of that. Yeah.
So I mean, as I'm just interested in this area because I've tried many times to get my records back from impossible, pretty much, to get it in an electronic fashion. And I have a background in data and analytics and so on, and it's impossible to actually get hold of your records. So I think the more needs to be done. I think probably to just get. I know I'm entitled to get the records through HIPAA and stuff like that, but in practical terms, it's just not possible. So more probably needs to be done to make the data accessible to the patients.
Yeah. No, it is possible. I mean,
it's possible.
I give you a mail and you do it tomorrow morning.
Yeah,
it's super easy. But no, it's not. So, and I take it as a suggestion. We will do even more effort to reach out and to understand that it's possible. And thanks for let's say knowing that the fact that you don't know it's possible is one lack that we have to fill. The clinical environment will get better. I come from a country, so I'm from Italy, where state-driven healthcare is not even close in a way to getting genomic information. United States gives you many options, and with both private and supported by your pay. So you are in an ecosystem that is probably one of the most advanced in the world. I'm sure you can find it. Other countries are catching up. UK is catching up, is more and more probable. In several countries, there's no sequencing facility at all. So that becomes kind of a challenge how to find it. In the United States, I'm sure you'll find a way.